I'm the token social scientist at the meeting. Feel free to throw things at me if you don't like what I have to say. So just to speak to the title of my talk, first of all, what's the relationship between regulation and innovation? Of course, some people tend to characterize the relationship as regulation is a block, an obstacle to innovation. Other people seem to often characterize it as something that lags behind scientific and technological progress and is always trying to catch up. But I would suggest that the example of the emergence of a regulatory regime for pharmacogenomics demonstrates that, in fact, regulation and innovation are mutually interdependent. It's a process of what we science and technology studies scholars would call co-production. I think nothing better illustrates that in many ways than the origins of the existing pharmaceutical regulatory regime. If we go back to the 1962 CAFALFA amendments in the United States, where did things like phase 1, phase 2, and phase 3, the need for randomized controlled trials come from? They came from academia and industry R&D. They'd been borrowed. For instance, phase 1, phase 2, phase 3 was something that was being developed in the National Cancer Institute. And I think what we're seeing now is exactly the same kind of dynamic interplay between academic science, industrial R&D, and regulatory science. So in this presentation, I want to reflect on the progress that we've made in developing a regulatory framework for pharmacogenomics, and I want to discuss some of the key challenges that remain, and to kind of highlight, illustrate some of those challenges, I'm going to use the example of warfarin pharmacogenetic testing. And I'm going to focus on the complexity of the governance framework, particularly something we've been touching on a lot today, which is the post-market regulatory space, the multi-level nature of that space, the multiple gatekeepers involved in that space. So just to give us a sense of how far we've come in the last 10 years or so, I want to start with 3 quotations. Now, the first one is from Jerry Collins, who was director of the Laboratory of Clinical Pharmacology. It's from an article in Nature Biotechnology. And this is, of course, in the midst of the Human Genome Project, lots of genohype around. And Collins' quotation illustrates the degree to which FDA and other leading regulatory agencies were still rather skeptical about the potential of genomics back in 1998. But we can see from the quotations from the current FDA commissioner and from this quotation from the EMA roadmap to 2015, the degree to which EMA, FDA, and other leading regulatory agencies have become very strong advocates for pharmacogenomics and personalized medicine. So how did we get from skepticism to advocacy? I would say that key to this process has been a process of building trust in pharmacogenomic data, building trust in the tools that generate that data, and building trust in the ways that data is then used in R&D and regulatory decision-making. And that trust-building process has proceeded through a series of regulatory experiments. Regulators have been essentially generating new knowledge about policy, which has allowed them to test their capacity to handle the new technologies and to gauge stakeholder reactions to their activities. And that's proceeded through 3 phases. So information gathering, then standard setting, and then implementation. So if we talk about information gathering, which happened between 2000 and 2004, what we see is EMA and FDA convening meetings very much like this one to bring together industry and scientists to talk about what's happening and to try and reflect on its implications for regulation. Interestingly, the FDA meetings were co-chaired by industry, had EMA participation from the outset. So we see 2 things. From the beginning, an attempt to create a harmonized regulatory framework between the US and Europe, and also a very close relationship with industry. Larry Lesko, one of FDA's most passionate advocates for personalized medicine, talked about a new openness and a new era signaled by these workshops and the ways in which they were organized. So this all led to, in 2003, draft guidance documents from EMA and FDA about briefing meetings or voluntary genomic data submissions, which created a safe harbor for industry to share pharmacogenomic data outside the formal regulatory space. Now, this is our first big regulatory experiment. Indeed, Janet Woodcock described the VGDS as an experimental space within which data could be shared without fear of consequences. This was the first time that industry had been invited to share data outside the formal regulatory approval system. So it really is quite a significant development, I would suggest. We also see, in terms of the FDA EMA collaboration, particularly around things like bilateral VGDS, the degree to which this emergent regulatory regime is being shaped through transnational harmonization and bilateral cooperation. Nowhere is that more significant than within Europe, because the degree to which the EMA has become the de facto regulator for pharmacogenomics and personalized medicine within Europe has simply underlined the degree to which its growing authority as a regulator within the European Union has been increasingly empowered through its role in regulating new biotechnologies, not just this one. I'll cite regenerative medicine as another example. So by 2004, EMA and FDA had established its role as advocates for pharmacogenomics, and this was made clear when both agencies issued reports in which pharmacogenomics was situated within a broader regulatory reform agenda, the FDA's Critical Path Report and EMA's Roadmap to 2010. Now, both these reports addressed the issue of pharmaceutical productivity crisis and suggested that the roots of that rose from the failure to fully exploit new developments in biomedical science, and suggested that a new product development toolkit was needed, including the use of pharmacogenomic biomarkers. I think this is also significant because I believe it's really the first programmatic statement from FDA and then from EMA as well about the regulators' role as enablers of innovation, not simply protectors of public health. Okay, so the second phase moved from information gathering to standard setting. We saw, so we'd seen with the voluntary genomic data submissions and the briefing meetings, the creation of a new pre-regulatory space. What we see in the process of developing standards through the Critical Path Institute and also the Innovative Medicines Initiative is the creation of a new pre-competitive space for the sharing of scientific knowledge between industry, academic scientists, and regulators. And the proof of the effectiveness of this new pre-competitive space came with the Predictive Safety Testing Consortium, where EMA and FDA cooperated on validating genomic data for use in regulatory decision-making. And this was, again, it's a first because it was the first ever cooperative decision by the two agencies made on the basis of a joint data submission. So at the same time as these standard setting initiatives were going on, regulatory agencies were also implementing pharmacogenomics in new product approvals and through the relabeling of established drugs. And when we get to this implementation phase, then I think we begin to see some very clear differences between the FDA and the European Medicines Agency, particularly in the area of relabeling. So the FDA very much took forward the idea of relabeling of established drugs as a way to get some early adoption of pharmacogenomics within clinical practice. So, for instance, it relabeled the cancer drug irinotecan to encourage use of UGT1A1 testing. But this effort by the FDA to push pharmacogenomics into the clinic through relabeling met with some resistance from healthcare professionals and healthcare payers, the kinds of things that Ron von Schack was talking about earlier this morning. We'd also had the approval of the Roche Ampli chip, the first pharmacogenetic microarray in 2004, approved by the FDA in 2004 for use of CYP450 testing. And we'd seen that product fail a series of health technology assessment evaluations in Europe and North America. So the diagnostics industry were in something of a quandary about where to go next, and they were looking to the FDA, so this relabeling process, as a way of driving adoption of pharmacogenetics. And by the 2005, then people were really hoping for some kind of game changer. And the game changer that people focused on was warfarin. Warfarin became the putative poster child for pharmacogenetics. And I think we all know about the problems with warfarin, about its narrow therapeutic index, the fact it's a major cause of serious adverse events, etc., etc. And Julia Kirchheiner, now what's her surname now, Ron? Stingel, who was with us earlier, sadly left, wrote this paper on personalized medicine, which really summarized the state of scientific knowledge. It pointed to the major pharmacogenetic component into individual variation in warfarin, suggested that that meant that warfarin was going to be one of the first example of CYP2C9 genotyping in clinical practice, but it also highlighted that there was a need for prospective studies evaluating such genotype guided dosing strategies. So a year later, the FDA's Clinical Pharmacology Advisory Committee voted 8-2 in favor of relabeling warfarin. The year after that, the FDA's Critical Path Opportunities list, its first priority was biomarker development, and warfarin was highlighted the need for pharmacogenetic-based rigorous dosing protocols. A year later, the FDA relabeled warfarin, but at that time it didn't provide any dosage guidance on the label. So I think there's a question there about what Federico yesterday described as a label which makes sense. And certainly there was resistance to this relabeling, again, from healthcare professionals because they felt it was inadequate to guide clinical practice. In the meantime, a study that was funded by the Critical Path Initiative, an RCT, was published shortly after the relabeling. And it did demonstrate that you could get to the stable dose faster with pharmacogenetics and clinical factors together, but it did not achieve its primary endpoint of a reduction in out-of-range INRs. So this randomized control trial did not provide the kind of clear-cut evidence that would have encouraged adoption by clinicians, and that's reflected in the fact that in 2008, the American College of Chest Physicians and the American College of Medical Genetics, as well as Blue Cross Blue Shield, a major payer, rejected warfarin pharmacogenetics. So at this point, the Centers for Medicare and Medicaid Services stepped in. They started a public consultation on whether they should cover warfarin pharmacogenetics. And in 2009, they completed their consultation, and they said basically more outcomes data was needed from randomized controlled trials. And in order to encourage that, it wasn't simply a negative decision. They said, we will pay for warfarin testing under coverage with evidence development if you're enrolling patients on a randomized controlled trial to look at the value of warfarin pharmacogenetics. So 2010, the FDA relabeled warfarin for a second time, but this time with pharmacogenetic-based dosage guidance, which was based on a study by Medco and Mayo Clinic, which was a prospective observational study. So it wasn't a prospective randomized control trial, but it was a prospective observational study. However, there was criticism of that study, and clinical uptake of warfarin pharmacogenetics in the United States is still limited. So what can we learn from the example of warfarin pharmacogenetics, one in which so much hope and indeed political capital of the FDA, I would suggest, was so heavily invested in these years? First of all, I think it illustrates the complexity of the post-market regulatory space, the degree to which multiple gatekeepers, health technology assessment agencies, healthcare payers, professional bodies, all have some role to play in the evaluation of new pharmacogenomic technologies and the degree to which they all have significant power in shaping clinical adoption and clinical practice. So Hans-Georg Eichler's presentation this morning talked about the different information needs and interests of different actors within this post-market regulatory space. I think the warfarin example illustrates that very nicely. I think it also illustrates the degree to which there are varying degrees of commitment to personalized medicine amongst these different gatekeepers. I talked earlier about the information gathering phase and the standard setting phases, development of a regulatory regime for pharmacogenomics. I think it's notable in those phases that the focus was on bringing together academic scientists, industry, and regulators, and probably there was insufficient focus on bringing together these other gatekeepers in thinking through how personalized medicine would enter the clinic. And I think it illustrates perhaps the degree to which this varying level of commitment to personalized medicine means that the kind of approach that's been adopted by FDA in its relabeling, which I would suggest is rather like what Hans-Georg talked about for drug approval. It's not so much learn, confirm, and label as learn, label, and confirm. The FDA have begun to see the drug label as an experimental space within which to investigate how you can drive pharmacogenomics into the clinic. I would suggest that it illustrates the degree to which that paradigm is not necessarily accepted by other gatekeepers. And it just illustrates the degree to which the authority of the FDA and other drug licensing agencies is primarily in the pre-market space. The FDA and the EMA can shape the direction of drug discovery and development, how it's carried out, because it has a giant yes-no switch in terms of whether or not it approves a product. Okay. So I think as well that we can reflect on the many topics on which people were agreed about around warfarin, but fundamentally the difference was an epistemological one. Effectively, the FDA was saying, if we understand the genetic basis of inter-individual variation and we have observational data, that is sufficient to relabel the drug. And clearly the people who were resisting that were saying, no, we must have randomized controlled trials. And in some senses, this is a conflict between evidence-based medicine and personalized medicine. And fundamentally, it's about the degree to which the randomized controlled trial, which is the gold standard enshrined in drug regulation, whose authority is in many ways inextricably linked with the authority of drug regulatory agencies, is now going to be adapted. And evidence-based medicine, I would suggest, is, as Timmermans and Berg outlined in their 2003 book, The Gold Standard, evidence-based medicine operates with an epistemology which is overwhelmingly empiricist, grounded in epidemiological and statistical reasoning. It's essentially interested in what works, and how the intervention works physiologically is less relevant. So I think this has some salience in understanding this epistemological divide and the conflicts that we're finding ourselves within. But this isn't just a difference between licensing agencies and healthcare payers or clinical professional bodies writing guidelines. The EMA's attitude to relabeling has been rather different to the FDA's. This quotation is from Reflection paper on pharmacogenomics and oncology. So for instance, FDA relabeled irinotecan to encourage UGT1A1 testing. EMA said that the data was suggestive but insufficient. So I think we are seeing differences, but they're not just ones about attitudes towards evidence. They're also differences in the institution's authority. So this again is from the same Reflection paper. Yeah, and it basically says that EMA doesn't have the authority, for instance, TPMT testing or warfarin, which were approved nationally, to relabel those drugs. Now, of course, we've heard yesterday from Peter, thank you, that in fact EMA's authority in this regard has been enhanced. So this is perhaps a historical difference that's going to change now that EMA has more authority. But the other difference between FDA and EMA is really about authority over companion diagnostics, and it touches on Fabio's presentation. So again, from an EMA guidance document, the legal requirements for in vitro diagnostics are outside the scope of this paper. Essentially, EMA has no authority to approve companion diagnostics. So when the Commission consulted on the future medical device regulations, it raised the possibility of a role for EMA in medical device regulation. And although there was some support for that, there was also significant resistance, and there is no real role aside from this consultation process for EMA in the Commission's new device regulations. Okay, so we've had problems with IVD device regulations in Europe, and many of these are gradually being addressed. I'm going to skip through this. They also in the States, the ambiguity about the status of laboratory developed tests has been a significant issue, which I think has hampered progress in clarifying the regulatory framework for personalized medicine. Fabio's talked about the new Commission proposals. I think it's a significant advance on the regulatory framework we have at the moment, but there are a number of areas of concern where I think we need greater clarification. I'm not going to talk about development in IVD device regulations in the States, aside from to say that the FDA, I believe, has drafted a guidance on how it's going to regulate laboratory-developed tests. That hasn't been made public. I think that's essentially a political issue, and we won't see that until after the presidential election. No, I'm not joking. It's true. So conclusions. So we've seen the creation of new socio-technical spaces, pre-regulatory spaces, pre-competitive spaces, new forms of transnational collaboration. The transnational collaboration is fueled by and reinforces the existing dynamic of harmonization and cooperation within the European Union and globally. But we also see differences in implementation which arise from different approaches to evidence, and also different legal powers. And we haven't seen any transnational harmonization on the device side of personalized medicine. And I'd suggest that's because the EMA has no authority over companion diagnostics. So there is no one for FDA to sit at the table with because of that lack of authority. So where do we go from here? So what do we do about health technology assessment? Can we have greater coordination between FDA, EMA, and other licensing authorities and health technology assessment agencies? Is there some kind of role for early health technology assessment where HTA agencies are brought into the evaluative product and the product development process much earlier rather than simply coming into the picture once a drug's on the market? I think EMA's authority over companion diagnostics has to be addressed. It's simply not acceptable that EMA can approve the drug and it can't approve the diagnostic. If we're serious about personalized medicine, then we have to address this regulatory lacuna. One way of doing that might be through the combination product route. The FDA authority over laboratory developed tests is, as I've suggested earlier, essentially probably a post-election issue. And finally, I want to leave you with a thought about a shift from voluntary to mandatory data submissions. Because if we're really serious that we believe that personalized medicine is going to transform healthcare, transform drug innovation, and transform drug regulation, then why shouldn't we move to mandatory submission of pharmacogenomic data? When the current regulatory regime based on randomized controlled trials and phase 1, phase 2, and phase 3, etc., were brought in the early 60s by the FDA, the agency didn't say to industry, well, this might be quite nice if you think about doing this. It said, if you want your drugs on the market, this is what you have to do. So I think we've reached a point 10 years on from the kind of early discussions where I think the science is mature enough, the regulatory regime is maturing, where we have to begin to have that discussion, even if that's a discussion that we're not going to complete anytime soon. Thank you.